Thanks, Salian, and thank you to the organizers for putting this course together. So my name is Anna Lebedeva. I'm a PhD student in Karandini and Harris Lab. And the goal of this session is indeed to talk about how you would go about tracking the same neurons recording with neuropixels probes across multiple days. Oops. So can you actually see my whole screen, or is this thing in a way, this black bar, is it on the way or not?
I don't see a black bar.
Okay, great. So the first question is why you would want to record from the same neurons for days and weeks with neuropixels. And the answer is that if you're interested in questions about learning or plasticity or aging or memory and so on, and you are interested in how specific neurons rather than whole population changes its response in these processes, then you might want to use neuropixels for that. They have specific advantages. For example, compared to two-photon imaging, you get higher temporal resolution. And also with neuropixels, you can access deep brain structures with minimal damage. And of course, you can record many areas at once. But there are specific challenges that come with that. So if you chose to use imaging for your experiments and you record on multiple days, you can just look at the images acquired on different days and visually compare them and basically confirm that you've recorded from the same units or maybe not. With electrophysiology, on the other hand, you, as a result of your recordings, get images like that have some wiggles. And they might look similar on different days, but you need some kind of algorithm to actually tell you which wiggles come from the same units and which ones don't. But even if you did have the same algorithm, the algorithm that would tell you which neurons are the same, it might be a good idea to first check the performance of these algorithms on neurons with known properties. And in some cases, this is possible. For example, when these properties stay stable over time, you can use these properties as kind of ground truths to first make sure that your algorithm works, and then you may apply it to neurons that do not have stable properties like that. So to some extent, this is true about primary visual cortex in mouse. So let's say, for example, that you've recorded 3 neurons on the first day of your recordings, and you showed many naturalistic stimuli to a mouse. And let's say that the first neuron prefers this first image, and the second neuron responds most to the second image, and the third neuron really likes this third image. Then if you have an algorithm to match these neurons to your recording on day 2, and the algorithm tells you that this is the match for neuron 1, this is the match for neuron 2, this is match for neuron 3, then what you would expect is that these matches have the same preferred images as your initial neurons. And now I'm going to tell you how we did basically that in practice. So we use NeuroPixels 2 probes, and by now you know that they have linearized geometry, which means that the channels are located directly on top of each other, and there are no gaps like there were in NeuroPixels 1. So this is important for our goal because the direction of movement of the probe is usually up and down as opposed to left and right. And therefore, if you record some activity on your set of channels on the first day, and then the probe moves in this direction, with NeuroPixels 2, you may still be able to pick up the exact same activity just on a different set of channels, as opposed to NeuroPixels 1, where you might lose some of this activity in between your channels. Moreover, NeuroPixels 2 are useful in this case because it's possible to select a set of channels that are distributed across the 4 shanks, and therefore they are on the same depth in the brain. And this way you can record with all of your channels in your region of interest, which is what we wanted. We didn't want the channels to be distributed across many different regions. And so what we did, like Celian mentioned, we used the most stable possible way to record. So we inserted the probe through a craniotomy. We then covered the brain surface with quick cast, and we then cemented the probe with cement to the skull. And we then also use this 3D printed protective cover to protect the probe from physical damage. We implanted the probes this way in 3 mice in left primary visual cortex. These recordings maintain good quality for quite a long time. So here is an example of some units from day 198 after implantation. And as you can see, these units still have quite good quality. Moreover, these recordings were quite stable over time. So here on the x-axis, you can see days since implantation, and on the y-axis, this is total firing rate in 3 mice, and it doesn't drop very much over time. And also the cluster count does go down, but not dramatically over time. But one thing that we noticed is that sometimes in between recording days, we have these drift events. So this thing is a drift map. Each dot here is a spike, and the color of the dots represents the amplitude of this spike. So the darker dots are higher amplitudes. And as you can see, the overall distribution of spikes across the probe, so this is the Y position on the probe and the X-axis is time. The overall distribution of the spiking activity on day 1 of the recording and day 2 of the recording is similar, but it appears that there is a vertical shift of this activity in between these 2 days. In order to deal with it, the procedure that we applied was to first stitch together the 2 recordings on day 1 and day 2 to make one giant recording. Then we applied a special version of Kilosort 2.5. And if you want to see exactly the details of that, please check out the paper. And then we sort these recordings together to correct for drift and to find matching units across the 2 days. But then, like I said, we wanted to check the performance of this algorithm. We didn't want to just trust it. We wanted to make sure that it actually picked up the same units. For this, we used a battery of natural images, 112 of them, and we showed them 5 times in random order. We showed them on the center and the right screen. We then looked at the responses of individual neurons. So here's an example, neuron 9, that responds equally to image 33, 69, and 90. And here's another neuron, number 19, that responds only to image 69 on day 14 from implantation, day 15, and day 24. We can then construct for each neuron its signature of its responses. So basically, here, each mark is a response to a different image, with red meaning higher response and blue meaning lower response on day 14 and day 15 for the same unit. And then we can combine these responses into these matrices on day 14 for all of the neurons and on day 15. And as you can see, these matrices, they look similar. In order to quantify that, we basically asked if a neuron's response is more similar to itself or to its nearest neighbor. So here are more than 200 pairs that Kilosort's algorithm told us are the same pairs. And if indeed they had, this unit 100 had most similar response to itself on day 15, then we would put a dot on a diagonal. And if instead it responded more similarly to its neighbor, then the dot would be off diagonal. We then make these plots for 2 recordings that are just 1 day apart. And as you can see, most units are more similar to themselves, with the exception of this 1 unit. And if the 2 recordings are 2 weeks apart, then still most units best matched with themselves, with a few exceptions. This is the summary of this data. So basically, we can conclude that if recordings are 16 days apart or less, then we can successfully track 93% of units. And if the recordings are 3 to 9 weeks apart, then still we can track 85% of units, which is quite high success rate. So to recap, we recorded visual responses in 2 different sessions. We then spliced together the 2 recordings. We corrected the shift in the data and we sorted them with Kilosort 2.5. And we then validated the matches that Kilosort 2.5 produced without access to visual responses. We validated these matches with access to visual responses. But I must say that nowadays there are even better algorithms to do this. So here's an example from Charlie Window, and you can find the code and some demos here using this link. And I basically was applied to 11 recordings that I made. And both Kilosort 2.5 and Charlie's algorithm, they find some jumps between recording days. But with Kilosort 2.5, you can see sometimes there is some instability. And if you then correct these shifts with Kilosort 2.5, you don't get optimal results, whereas with Charlie's algorithm, it looks much better. So to conclude, Kilosort 2.5 has the ability to track same neurons across recordings, even if there is probe drift between them. We validated the result of this algorithm using visual responses, and if your recordings are 16 days or less apart, then you can trust this algorithm to do well. And nowadays, there are other algorithms that may be interesting to try and compare. With that, I would like to thank all my collaborators, and thank you very much for your attention.